Today we have Kim Branson, the SVP and Global Head of AI and Machine Learning for GSK. So Kim, thank you so much for being here. Welcome to Raising Health.
Thanks, Vijay. It's great to be here.
So maybe the one place to kick it off is that I think people would love to get to know you. And, what got you interested in this space in general? Like, my guess is like as a kid, you like computers, but you must have also like medicine and biology. Like, how did this all start?
I actually never done any biology, never even thought about it until I actually got to university. And then I discovered molecular biology and then bacterial pathogenesis, and you have this interplay within the immune system and bacterial stuff, and I was sort of hooked. And yeah, you're right. Obviously, as a kid, I had played with computers and liked computers, but I was far more like a math, physics kind of kid. And I sort of, once I discovered this whole world, I was sort of hooked. And then I discovered it wasn't just sort of blobology, there was like structure, right? So then X-ray crystallography and things. And once you sort of put all those things together, it's a sort of a very seductive thing to come into, to like, how things really work, right? So I was sort of always drawn by the allure of sort of structural biology and like, we love all those sorts of things. And then I realized like between those long hours of during incubations and things like that I could just go and do this instead. Right?
And this being computation.
Yeah, doing computation and doing small molecule drug design. I mean, when I had sort of finished my PhD, even during my PhD, people were like, well, computational drug design, does that work? And the answer is, well, yeah. And it definitely is starting to work more, right? And so the people I did
But you're talking about like, well, that's like 20 years ago.
Yeah, so we're talking like, yeah, it makes me feel old. This is like, this is like 99, right? Yeah. So I was working with Joseph Agis and Peter Coleman and Brian Smith, and these are the people behind the first neuraminidase drug, which actually was designed with computation. So they used a program called Goodford's Grid to map the protein surface and work out to put this functional group there. And it was all based off the original sugar molecule, but it was actually designed using a computational method. They were believers in it because they had built something, and it became marketed as Relenza, I think, by GSK, ironically, where I am now. So yeah.
Full circle. Actually, after that, you went on to go to some startups.
Yeah.
And also to go on to pharma. Curious to get your take on one versus the other, and how you see that ecosystem.
Yeah, I mean, I did spend some time at Vertex, and ironically at Vertex, like in biology, as like the best model you can build has like a figure of merit of accuracy like 0.6, 0.7. If it's 0.9, anything that looks great, there's something wrong. And this is sort of this is pre-Bing. This is the ideas of PowerSet and Bleco, if you remember that, and a whole bunch of search things. So some friends of mine were building a search engine. And so I went to sort of join them because I wanted to play with large data sets, right? And I learned a lot about doing machine learning and things on very large data sets. And that was really exciting because, the ups are up. There's not many encumbrances. You just get on with things. Mm-hmm. But then you're inhibited by sort of scale and capital and everything else and those other types of things. Whereas at a large company, there is a lot more capital, but there's also, it's a very big ecosystem. A lot of people doing various things. The trick is how you direct it, right? Mm-hmm. And so they have different sort of strengths and weaknesses. And for me, it was, I guess I could bring what I learned from one to the other, right? And then back again. But I sort of, once I did, started in the startup sort of world, I sort of stayed in that bit for, 3 companies. For me, I still really like that feeling of pace and urgency.
Yeah, in
right?
the startup.
Yeah, and you could just, if you carry that through, I think in a large company it's very easy to sort of sit back and go with the ebb and flow, and you can do that if you do it today. If someone else delays 3 days, something else it doesn't delay like a month at the end, right? So like do it now kind of thing. Energy of startups, and you recruit a different type of people and things like that.
So what drew you to GSK? Like what brought you there?
So GSK was interesting. I was at Genentech at the time, and I had a lot of friends at GSK, and they were wanting to do AI and things. They were asking me, how should we organize this? Where should we put someone? They were dead set on a particular location. And at the time I was like, you're not going to find the people you want there, and you should organize it this way. And eventually, I was quite resistant to going to GSK. Because of, outside perceptions, and you see the same people at conferences that for the last 20 years complaining about the same things. You're like, I won't work there.
But it's right, the big company thing.
Yeah. And so I eventually they're sort of like, well, why don't you just come in and interview? And so I went to interview, and I sort of met Hal, and I met people, and I realized actually it's kind of very, very different, right? Then realized that it was in a phase of this turning itself inside out. So these big companies don't reinvent themselves very often.
It's hard to do. Right?
Yeah, but they were really doing it, and they were really serious about doing it. And so, I was sort of meeting Hal, and I knew, Hal, through Daphne and other people, and you knew that he was, and he could write some Python, right? So you got a CSO. Yeah, he got a CSO. He's actually an engineer by background as well, so who can really write code. And I realized, this guy has actually worked with machine learning people. He knows what it takes to do things, and they're fully investing in the things I thought were very important at the time, which was, these large genetic databases coming online and functional genomics. So he was way ahead in sort of the thinking about CRISPR and the gene perturbation to generate data, and saying there's going to be explosion data. You can see the trend, so we're going to need machine learning to make sense of it all. And so they weren't sort of just dabbling in we'll have one group and we'll do some contracts. We'll establish an entire thing, and this will be core to our strategy.
Wow.
So I thought, well, that seems like an opportunity. I guess I was thinking at that time of doing another company or something else. I thought, well, worst case scenario, I'll just get another job. It's not very hard. And, interestingly, there was a book running, you probably know, but people were running a book that how long I would last there, 2 weeks,
oh, really?
6 months, 2 years.
Okay.
So I beat the odds.
Yes, wow.
It's 5 years this Monday that I've been there.
You got a short squeeze on them.
Yeah. If I knew, I'd know. Yeah. So I've stayed, and I think we've built a lot. I figured, I know how to do things in smaller companies. I know how to build things where you can attract the people and everything else. But the next sort of leadership challenge is, how do you take a larger organization, create something within it, but also bring everybody along for the ride and things like that, which was a new challenge.
Yeah, well, so how do you do that? I mean, that's a great topic.
Yeah. So I think I guess Amdahl's law isn't just for CPUs, it's for companies. So that ratio of computation to communication, right, is really important. So what I've realized is sort of, you do have to, you have to spend a long time explaining what seems intuitively obvious to you, to a large organization, because you've got a market of very busy people. They've all got jobs to do.
And they're coming from different backgrounds too.
And they're coming, and drug discovery takes people from so many different disciplines together, right? Plus manufacturing, marketing, it's an entire thing. It's as complex endeavor as anything else. And so they don't all have the same background and training, and also different ages, right? Right. Technologies change a lot. So you've got people living in different stages at what they're experienced with. And some of them are skeptical. We've got something that works. Does this really work? And compare it to things. So you've got people that are, it's very classic innovator's dilemma. Well, it's not very good now. And I'm yeah, but I have to explain. Think of the rate of change.
Yes.
Right?
We got Moore's law.
Yeah. And then you've got true believers as pockets of those things. So what it is about kind of convincing people, doing a lot of messaging. But to a certain point, I discovered, you could talk a lot, but then you should just shut up and build for a bit. So we just basically came in and said, no, we're not working on those sorts of things, create a little sheltered bubble, built an organization, built a whole bunch of capability. And then you're at this phase now where it's sort of the installation phase of the technology, right?
Yeah.
Where you're now going to plug it into, where you've done these little pilots and things, and now you want to sort of actually make it part of the process.
Yeah.
But I think that's where you have to communicate, right? And it takes a long time because it's also geographically distributed, time zones, things like that. So you'd be surprised at the amount of time it takes for a message to percolate. But eventually, everyone gets behind it. Then you've turned the ocean tank, and then it's this unstoppable juggernaut, right?
Yeah.
Because there's a lot of capital and data, and
that's the strength of a big pharma. You've got all that, right?
Any discipline you want, you can find an expert.
Yeah.
And if they don't know the answer, they know the expert who does.
I think the question now is okay, we see AI around us, but how is it going to affect drug design? And I think what's really cool about what you're doing is that you're doing this today. I don't know if you could give some examples.
Yeah, so I think everybody thinks, if AI is successful, you'll enter in the disease and it will say this target and pop out the molecule, will make it, and we won't even need to test it. We'll test it in virtual humans, right? I always roll my eyes when I hear that.
Well, and maybe that'll happen, but maybe that's 50 years off.
Well, yeah. So I think what we're seeing is our group works all the way across the process, right? We have an enterprise focus, but the first thing is what to make the medicine against, right? You could design the best drug in the world with the best pharmacokinetics and safety profiles and everything else, but if you're modulating the wrong thing, it's not going to have a clinical effect.
Right.
So the first thing is it's the target. Pick the target. So we have these large genetic databases of people sequenced and their medical records and things like that. And we look at here are people with a disease and here are people that don't have a disease. And so what's different about them? And they have lots of differences, but what differences are really significant? And then are they driving the disease? So you might, first of all, do people that are diagnosed or not diagnosed, and that's one way of doing things. Or we can use machine learning again. So we use machine learning again on clinical imaging. So you have a bunch of people that may have a different range of, say liver enlargement or scarring of liver or kidney function. And rather than having a person score that by hand, we can have AI model go and do that. That gives us what we call a continuous trait. Then we can take that information and do GWAS, right? Which is the way you'd work out what's different.
Genome-wide association.
Yeah, exactly, right. And now we have some other variants on that we're interested as well. Like, but once you have that, you then need to work out what does this genetic variant do, like biologically. And the first question is, well, what cell types is it acting in?
Yeah.
Then it could be like several genes away, right? So you want to predict which is which, where is it acting? And then it is, okay, well, what is it doing? Is it making more of the messenger or less of the messenger or changing splicing, right? So you get a different form of the protein. And so we have methods they're machine learning methods that will predict the directionality, right? And so that's in use in production today. That helps us interpret the variants, right? And so that helps us idea, okay, this thing, make more of this protein, these people seem to have this disease. Therefore, this gives us a clue on how to modulate the disease, right? We have a lot of different methods for looking at cellular imaging. So think of things like recursion, things like that. Predicting the effect of a small molecule, or looking at a cellular phenotype. We have active learning systems for discovery and doing experiments in biological models of disease. So rather than making a small molecule, tool molecule now, we can actually just perturb, we just turn up and down the gene, right? And you can do pairwise as well, you can do more than one. And using rather than CRISPR, using something called a TALEN, we can turn up or down these things as well on a continuous basis. So we can go from like small amounts to large amounts of protein, or turn it down slightly and see what happens. So you can modulate things like that. And so we have a whole active learning system where we feed in, here's our data from, here are our hypotheses from genetics, here's our stuff from the literature, here's like, just trust me, I know kind of stuff. Because you get that in biology, people have experience. Yeah. And we can't do a genome-wide screen, just try everything. So we use this model that says, okay, I want my RNA expression profile to look like this. I like some proteomic thing, or maybe a functional assay to look like this. And I also like it to be not toxic and a target that's tractable, that I can make a molecule against, right? So you can bake all those things in, and it will do a round of experimentation and say, well, here's information back, right? And then based on what I've learned, I might say, and based on my connectivity understanding, I might say, okay, I'm going to walk up there and mutate this guy because it's the common ancestor of these two proteins, right? And this is how the signal transduction works. That's about 20% faster, we've shown, than doing a random screen. And as we go into the clinic, we use machine learning we have computational pathology. So we've been working on computational pathology for a while. We have even a GSK professorship of computational pathology, a guy called Nasir Rajput, who works with us closely. And we use that for, first of all, we do a lot of things where people say, is the target expressed in the tissue, particularly in oncology, right? And that's just, you're staining it with an antibody, and a human pathologist looks at it and basically gives you, I think, about 63% of the slide is this, and you give it to a few different people and say, yeah, it's expressed high, medium, or low. Yeah. Well, with machine learning, I can tell you how much it's expressed, because counting brown pixels is pretty easy. Right. But also I can tell you which cell type it is, right? And so then you can, obviously, free the pathologist up to do other things. But then once you've got that, you also can say, if you get response data from your trial, you can say, well, now which cell types are expressed tend to respond more. You've got more data, and you sort of iterate on the model. So there's things like that. And we did a very heavily instrumented phase 2 trials. We measured a lot more proteomics things and everything else than before. And it's very expensive, and everyone's sort of like, well, how do you know this will be useful? Like, no. Most of it won't, but some of it will be. And we were able to look at that and analyze that data and find this subset of people that we know if we lower their viral surface antigen to this level, they are basically functionally cured, right? And we know which group it is. And now you go, okay, now I need to go and find a medicine that brings them to that state, and I can treat them with this drug, and we're good to go.
Well, so for the audience, could you give a sense for the delta between what you can do with ML now at GSK versus maybe the way this would have been done 10 years ago? And the delta in terms of time or dollars or whatever.
Yeah. I mean, certainly for some of these things, you would have to do a lot more experiments, right? So the genetics, right? If you didn't know which way the variant was, well, you'd have to go and engineer a system and make a model system and turn more of it up and turn more of it down, or overexpress it or not, and then make a whole bunch of surrogate readouts. A lot of these things about making continuous traits for discovery, well, unless you got a whole bunch of radiologists and scored a whole bunch of things manually and did it all, right? Yeah. Would take a long time, because that's a very easy parallel cost, right? We can calculate. The complexity of some of these sort of things is, you can't go through a very small N high D space and try and find out some correlations of something that you just
can't do that many experiments.
No, and to do it reliably, and some of these things are very complex, right? The Hep B example is, it's based in like an autoencoder type thing, but you're doing a lot of feature compression type things, right? And so it's interesting because AI has become so useful because we've got so much more measurement technology, right? Cheap measurement technology. So we can do, we can do generalized sequencing cheaply. We can do RNA-seq. We can do the single cell type stuff. You've got this data. You've got more measurements now, but you can't do anything with it unless you've got the machine learning. And my central hypothesis has always been medicine has advanced as new measurement technologies come along. And as the measurement technologies get cheaper, we learn more. So if you think about cardiology, right? Swan-Scott's catheter, measuring things in the heart. Not very people can do that. The story of the first guy to do that is bonkers. He did it to himself.
Really?
Yeah. It's like, I'll just do it to myself and not pass out. Absolutely crazy.
All the old school stuff is like that too, by the way.
Yeah. It was, the guy, the Helicobacter pylori guy.
Yeah.
Crazy Australians. Like, I'll drink it. I'll see it. Like, and they at the time they had a hypothesis it was bacteria. They weren't actually sure.
I know.
They were like, I will try business and whatever else. Then we'll hopefully clear it out.
Yeah.
But I think they got a Nobel Prize for that.
I believe they did. Yeah. So it was worth
self-experimentation.
Yeah.
Don't try this at home.
Yeah.
But from that, you've got blood pressure cuffs, you've got things, and now we've got these, right? Continuous pulse measurement, things like that. Yeah. So as they got cheaper, we learned more about cardiology, right? And then that led to discovery. So we've got more measurement technology, but it's in such high dimensionality on people that I can't make sense of an array of like a whole bunch of expression changes which just fluctuate around baseline versus a disease patient.
Yeah.
Right.
Yeah.
Like who can make sense of that? So we could generate it. We would, if without machine learning, we wouldn't know what to do with it.
Yeah.
Right? We'd have to do various sort of linear things and take out the most obvious stuff, which might be wrong. So yeah, I think it's hard to compare, in the event of the data aspect.
Yes.
Right. The change things. Yeah.
Well, so I'm curious, a lot of our listeners are people that are doing startups or in the startup ecosystem. Like, what have you learned, would you take to a startup if you were to start one now?
Yeah, I think if it was in the drug discovery space, I mean, even just thinking of like AI stuff in general, and it's about data. And it's also about unique data. So what you want is, I think a common problem I see is people try and build something as like, well, they don't have, here's what I want to set out to do. And like, well, we don't have exactly that data, but we've got this sort of data. So if you solve this, it's probably good. And so people forget that the data they've got is sometimes a proxy for the real thing. And what you should say is if that data doesn't have the answer, you should go and generate the data. And actually be able to generate the data is going to be critical because you're going to run into the thing at some point, you'll run out of it and you'll need to generate the data to build a better model. Right. And that also becomes a competitive mode.
Absolutely.
Right, because you'd have the stuff that you can generate plus the public stuff that you've got, you're bringing twice data to the page.
Ideally, someone pays you to generate the data.
Yeah, or gives you the data and then pays you to buy the model back.
Yes.
GSK won't do that deal with you, just to be clear. But yeah, the data aspect is key, and they generate more data, right? Like invest in that, because honestly, more data with a simple algorithm is an easy sell, easy install.
Right.
And like data wins, look at the Netflix challenge. How was that won? Bringing different orthogonal data to the table, not with a smarter algorithm, right? Yeah. So I think that's a key lesson, is inventing that, and then think about the ML and the fancy algorithm and things like that. So one way I look at a lot of companies that come to us, I look at like, what unique data do you have?
Right.
And what's your ability to generate, and what scale do you have, right? That I don't have, for example. And if your pitch is just like, oh, we've got smart people, give us your data, we'll do cool stuff. I'm like, well, I've got smart people too. Right? That's not going to happen.
Yeah. Well, then what differentiates the data in your mind?
I think is either you've got it's uniquely suited to understand, so it's clean, right? You've got less batch effects. You can generate more of it, and you can control the sampling aspects of it. I think those things are key. Could then basically understand how your method might behave under different scenarios, which will reflect what people may have, right? Because there are some biases in their systems, for example. I'm just thinking here in the abstract. Those are kind of the key things you would want to kind of pull in. And then when you've got that, then the ML thing doesn't really matter, because it's honestly you're selling the solution at the data stores, and people don't care whether it's like, the mechanical Turk type thing or whatever it is. You can sell a random forest. If you trained a really cool random forest, it doesn't matter, it's not a neural network or whatever else.
Oh, and sometimes
random forests,
that goes a long way.
They do go a long way, right? They should be the baseline everyone should start with for everything, right? And you should always compare your information gain above a random forest. That could be depressing for a long time.
Yeah, or either that or any of the other simpler methods. Even just a single layer neural net.
Yep.
Which is logistic regression, basically.
Yeah.
I mean, that is the same thing. It's funny, actually, how I think that will go full circle, by the way. There's all this love of the complex stuff, but we'll see what happens.
Yeah, I think people will realize that, there's this great paper by David Hand, right, which we may have shared a long time ago, right, called Classifier Complexity and the Illusion of Progress. Mm-hmm. It's very old school ML where he looks at some UC Irvine stuff, and for them the sophisticated method was like an SVM, but it's like Fisher linear discriminant analysis, a bunch of things, and these very toy data sets, but showing that like basically the simple stuff gets you there, and for the extra 5% gained, you need all the extra complexity. Yeah. I think that is still true now. I think the other thing for startups is work out how good the thing needs to be before you start. So talk to your market, work out like if it could do this, do these things, yes, I would use it. Yeah. And then go and work out to do that. Don't build the thing and then say, well, I've got that. Yeah. Because then they'll have to work it out, and maybe you're far off, or maybe there are some criteria, right? And that way it stops you, you know what to stop and when to start shipping. And
But so let's say a startup had the differentiated data.
Yep.
And what would you want to see in the algorithm to get excited?
I want to see well-characterized robustness and reliability criteria, right? Don't give me just point estimates. Give me a point estimate with some measure, right, of confidence and error, right? Like you got to have that.
And even that's actually tricky to do sometimes. Yeah.
I still think there's a lot of machine learning papers out there. It's like, well, my thing's better. Here's my table. Here's my stuff in bold, which is naturally better than my stuff. Yeah. And by the way, it's different at the third decimal place. Like, right. That's the other thing is like for a lot of people's stuff, the 10% difference, they're like, I don't care. It's not worth the cost of me buying it in stock.
Oh yeah. No, for like 0.1 in AUC, you're not going to.
No, you want the precision curve, right? Is really where it's at over AUC, but you got to work out what you need to be at to make it meaningfully different, and then how well is it engineered, and then can I plug it into my system? How does it work, right? How the installation stuff, because often there's the people who use it in a company, and then there's the people who have to operate it and run it, and how do we control for model drift and everything else. And so you're selling to a user, but the people who are buying it and installing it that are also part of the decision process are like the engineering stuff. So you got to think very carefully how you make it easy for them to use. And I would there can be a lot of people like stick it in the cloud, stick it in my cloud, let me run it on-prem. Like you have to think of different ways to bring it to the market. And that depends on the industry. But like the more ways you have it that make it easy for people to integrate and monitor and watch, and it's secure and you've done all that sort of stuff, the easier it is for them for it to be a frictionless thing. Yeah.
I know you still like coding.
Yeah.
and I know you like playing with AI. So like what have you done with your own hands recently? What have you been building?
One thing I built recently is I get all these emails, reports, right? All these various types of things. And like, I usually have one question I want to know. It's one thing. And it's like, I can't wait to do it all. So I realized like, I just think that like dump some these emails, because they want the sort of standard title, onto a folder on my laptop, and I run a little LLM that has my questions I want to know, and then it writes an email and just sends it to me, right? So like, that's one thing I built recently. I'm pretty interested in, the long context windows sort of stuff versus like, do you need a long context window for task planning, or do you need a whole bunch of specialist tuned LLMs for things?
Or agents for
for
going through the search.
Yeah, no, and I think search has fundamentally changed because what isn't like used to be like find me the document and I'll read it. Now it's just like, here's my question, you read the document for me and tell me my answer. Right. The initial version of this LLM for reasoning over multiple scientific documents is actually something I wrote myself. Right, and I wrote the prototype and put it together, and now it's so I think you still have to kind of build things, and I think also it keeps you thinking about aspects, and I also think it also helps you know about your infrastructure as a company, right? If you're kicking the tires on it regularly, or if you find it difficult to do something, then the other people do, it's probably slowing them down. So the thing I think a lot about is how do I speed up the iteration cycles of anyone across GSK, right? So not just me writing code, but people to access data and get data and think about things, right? Like if it takes them 3 weeks to get a thing to answer a simple question, then that's going to multiply down the chain, anywhere after another quarter or 2 quarters before they've done something, right? Yep.
Okay, so let's say it's 5 years from now, and we're doing another podcast. Hopefully we do one sooner, but like let's say it's 5 years. What are the examples that you're excited about that you've done? Like obviously that hasn't happened yet, so I'm trying to think like what do you think? Where do you think you'll get?
I think we'll have what I will call computational methods So every software, every drug GSK will launch will have software that sits around it. I think that's going to be true of everybody. We're going to see there's software that will sit around every drug, and we'll say, who should use it, what they're likely to be at baseline, more accurately what they're likely to be plus one cycle in drug, because it's very difficult to predict people's responses at baseline. Maybe by then we'll be better at it. I think we will know a lot more about IO. I think we're really understanding how this is immunotherapy. GSK is very much really actually an immune programming company, right? Vaccines are ways of programming the immune system, right? And we're seeing a lot of different vaccines. I think we will understand really the whole thing about IO is like 20% of patients respond, right? And it's an amazing response, but which 20%? Right. We'll know a lot more about autoimmune disease. So I think we'll see a lot more aspect with software, but I think also the age of single cell and perturbational genomics is just getting started. Yeah. Right. Like GSK has been generating exponential data growth, right, since I started. And we are generating large scale perturbational data sets purely to become like either a lookup table, so I don't have to do an experiment, right? But more importantly now, an inference table. Can I infer to an experiment, right?
Kind of like the Human Genome Project becomes a lookup table.
Yeah, exactly, right? So you're building those sorts of things. So I think in 5 years' time, what will happen is I think we'll be doing fewer experiments, but more informative experiments. But I think also the loops into observational cohorts, so we measure things in people where we're not changing management, we're just learning about the disease, right? We're literally running trials to learn about the disease to add to the public-private kind of mix of data. But also, so I think that's going to be full scheme. Everyone will be doing that. But I think, this whole weird thing of disease heterogeneity, we'll actually start to really understand what that means. Right. And also kind of...
It won't be so mysterious, it'll be decipherable.
Yeah, I think we'll know a little bit more about what's happening. And this is where I think, interestingly, ML will start to collide with really the kind of mechanistic modeling of biology, because we know a lot about medicine. We know organ systems and things like that. We don't have to start with just gene expression in the thing and tell our algorithm nothing about, by the way, this is the person, this is how things interact. We can have these sort of structured priors. So, I think this is where that sort of systems medicine approach will become very useful as well. So we will have a lot more models, I think, that'll be used in place for discovery systems now as well.
So what do you think is sort of the rate limiting step in getting, advancing this? It could be people, it could be money.
Well, it's like the trite answer is data, but the real answer is data with outcomes, right? So like I can generate all the perturbational data I want, but what I really want is like, samples from people, healthy people, samples people with disease, but also want as what happens when what's a clinical trial? We're perturbing someone, right? With a therapy. We're seeing what happens.
And clear causation.
Yeah, and I think that's where we need the outcome data. So outcome data is the gold standard data to have, right? It's the rarest data. That's really like one of the advantages pharmaceutical companies have, is they can generate data in humans at scale in a safe, ethical manner. That data, plus if you measure all those different things, we start to instrument these trials more. To me, it's a complete waste. If we drag in 200 people and we monitor them really closely, we should measure everything we can, right? And bring that back. And there is a patient burden and you can't overload them, and there's always a tension between that. And obviously we should do that in the right fashion. But that data with outcomes, that's the rate limiting step, because what we did to them, you've all measured the stuff, and you know how their disease progressed and what happened to them.
So how can we sort of minimize that bottleneck?
That's where I think that we either need to create larger public-private consortia or public thing. Like, what everybody wants to do is like, oh, can I have all GSK's clinical data for this trial? And I'm a different company. I'm like, no, of course you can't, right? Like, I should hand over my unique competitive advantage to you. No, I'm using it right now to develop things. But what we actually need is, I think we need to do large-scale observational cohorts. And the problem, and these things exist now, but the problem is what they measure is limited by the money they had and the hypothesis they had at the time. But usually you've gone and looked at something else and like, oh, I've got a new thing. How do I measure that? Well, we need to bank samples, right? And sometimes you want to bank the samples and kind of invoke Slacker's law. Like, it'll become cheaper and better to analyze these.
And that usually is true, right?
Right? It's very true, right? And sometimes, you can't do it with frozen tissue, but also we should be establishing, like, it's not just genetic sequencing and electronic medical records. It's genetic sequencing plus samples, but also we need to be following people over time. Like, we don't even really know what the immune system looks like over time. Right? It's kind of bonkers. So that's where I think that's the rate limiting step. And then you've got treatments and you've got outcomes, right? For those people. If the machine learning community or the drug discovery community in general had more of those sorts of things, right, I think we would be in a better place. And I think that's what's going to come, right? But that's actually what, honestly, I think is the work of, government and industry to fund those sorts of things. And maybe there's a time exclusive access and things like that. But eventually, if those become public resources, they become a common good. But I think if you look at where machine learning has had massive strides, is when there's cheap way of collecting data, right? So web search, image network, sorts of stuff, right?
Yeah.
We're good at those sorts of things because there's data sets. We as a company, we do put out data sets of machine learning challenges at Europe's and things like that. So we've done, the gene disco challenge for perturbations. There's a few of them. You can see them on GSK.ai, and we put out a prize and things like that. You can make real money.
Kim, thanks so much for joining us on Raising Health.
It's been a lot of fun, Vijay.
Thank you.
Thanks.